Hello everyone, good morning. This is a small room, so we can talk without the mic. Everybody can hear? Yes? That's great. It's nice that we could, that you so many are here, and this is going to be very exciting. It's really a true honor to welcome today a distinguished speaker that is Professor Dame Angela McLean. And so she is a mathematical biologist, and by training, and then she also has been helping transforming our understanding of evolution and spread of infectious diseases. And you're going to hear about this science that she has been developing. So Dame Angela has served as the UK government chief scientific advisor since April of 23, and she also worked as a professor as mathematical biology at Oxford University and as a research fellow at the Institut Pasteur in Paris before. So Angela pioneered research in infectious diseases modeling and has provided invaluable insights in how infectious grows within individuals and spread among populations. Through her career, Angela has been a passionate advocate for evidence-based policymaking. So she has consistently worked to bridge the gap between science research and practical applications, highlight the vital role that rigorous science plays in informed decision-making. And of course, this is something that is always very important when we have deep research results and how to really make it also understandable and used in society. So her contributions to science and public health have been widely recognized, of course, and she is an elected fellow of the Royal Society, and she has received several prestigious awards such as the Gabriel Medal and the Weldon Memorial Prize. And today, Dame Angela will share her insights and on the theme of from mathematical biology to engineering biology and bridging academic research and science advice for government. Drawing on her vast experience, she will explore the intersection of scientific research and public policy. So highlight the impact of academic discovery on societal well-being. And she will present this scientific talk and for some 15 minutes. And after that, she hopes that she has engaged you all in lots of questions for another 15 minutes. So please get ready for that, and we hope to see a really engaged half hour here in front of us. So now please join me in welcoming Professor Dame Angela McLean on the scene.
Thank you so much. So thank you all very much indeed for having us here to visit at OIST. It's just fascinating to see this big mixture of people from all over the world working here in such a beautiful part of Japan. I was told very firmly that they don't want to hear bureaucracy. And of course, I work for the government now. I am actually a bureaucrat. It's really hard to say those words, but that is true. That is what I do. But I was told, no, come and talk about your science. I thought, oh, yeah, I used to be a scientist. And so I thought what I would do is present two examples from my own work. One is actually from my own PhD work, is a piece of epidemiology. So it's thinking about the spread of infection amongst people. And then the second is a piece of work about what happens inside individual people. And then in the third of my 5 minutes, I thought I would fulfill the promise of the title and talk a bit about how I came to think about how important engineering biology is for the UK government to understand about. So the first one is about me when I was about your age. So I had done an undergraduate degree in maths. I'll tell you a secret, I hated it. Because the reason I'd gone to university to read maths was because I really loved solving problems. But even in applied maths in university, or at least in the university I was at, it's not about solving problems. You would get as far as the proof that a problem exists, and then your work is done. And I didn't like that. But I think I was very well advised to stick with the maths degree. They said to me, you know, it's just a very useful passport. You can do more or less whatever you like afterwards. So I stuck with it. I didn't have that joy of doing an undergraduate degree in something I loved, but I stuck with it. And then I was very lucky. I was made to do a course that was called Differential Equations. My personal tutor said, no, you must do this course. I said, no, I hate differential equations. But she said, yes, but it's really biology. So I was taught by an absolutely fantastic scientist and teacher. His name is Jim Murray. He wrote the sort of most standard textbook in the kind of mathematical biology that I would say is really about expressing in equations how you think a system is working, and then using those equations to try and understand the world better. So my first example is actually from my PhD. I was working on the question of, in a country where the birth rate is very fast, at what age should you vaccinate against measles? And this is a problem because you can't vaccinate a newborn baby against measles because the newborn baby has many, many antibodies derived from the mother that mean the vaccine won't work. Those antibodies will just wipe up all the virus in the vaccine and there'll be no immune response. In wealthy countries, you wait until the second year of life to vaccinate because then all those antibodies have waned. You can get a good immune response from the vaccine and make the baby immune. But of course, if the average age of infection is, say, 1 year, you can't wait for the second year of life to vaccinate. So I was making quite complicated models. They were partial differential equations because we were very interested in how things change through time, but we needed to keep track of age for these children. And so I was writing code. That was basically what I was doing. And I would sit in London and run this code. Actually, it took so long to run that I used to knit whilst I was waiting for it to run until somebody taught me how to make a batch job. And often this thing would happen. So it's well known. So we're going to have, because we're talking about epidemiology here, so this is going to be time sort of in years, okay? And this axis will be number of cases. You happy? So this is just number of people, usually children, with measles. So what my my models would do is they would produce, I mean, of course, they were beautiful surfaces. They were lovely, lovely things because they were grids over age and time, but I can't draw that. So what we would have is these epidemics every 2 years, quite regularly, until you started vaccinating. You vaccinate here. And what I would find is that initially things would be brilliant. The disease just melt away. Even if, according to the theory, I wasn't vaccinating enough to eradicate. And then nothing would happen, nothing would happen, nothing would happen, and then boom, it would come back like this. And I thought, oh, how weird. Why is my model doing this funny thing? And then I got married, and we had no money. So for our honeymoon, we visited the Gambia. So the Gambia is a tiny country in West Africa where there is a big medical research council research station, and also Save the Children, which is a British charity for children's medicine mostly, has quite a big outreach. So we went to the Gambia because we knew various people there as a work trip. And we got there, and they were all in a big panic. They were very, very worried. They'd had a great measles vaccination program. They thought they'd eradicated, and they were getting cases. They were getting cases amongst children, and the records weren't really good enough to know if those children had been vaccinated or not, but they were desperately worried that something had gone wrong with their refrigeration, because measles vaccine has to be kept cold to remain active. So there was this. They were roughly here. They were about here in this process. So they'd had bad measles. They'd introduced a good vaccination campaign, but not enough to eradicate. So after a few years of really no cases at all, measles had come back, and they didn't understand why. And I was too junior at the time to say, oh, I know exactly what's going on here. But I went back to London and stopped thinking about this as an artifact, went and read the literature, realized that this is actually quite a common pattern. And that what is happening in here is basically a shift in a dynamical system where you're going from everybody being immune because they've had measles, to what you would like is for everybody to be immune because you've been vaccinated, but you're not quite getting there, right? Because you're not meeting the threshold that would eradicate. But temporarily, to start with, you get this extremely low couple of years where you think you've eradicated. So this got named somewhat cheekily, sort of as a joke actually, as the honeymoon period. And these as your post-honeymoon outbreaks. They're not actually as big, they're much, I've drawn that wrong because they're smaller and a bit more spread out like this. And that's now a well-known phenomenon. These are called, that's called the honeymoon period. These are called post-honeymoon outbreaks. And it's a well-known phenomenon in epidemiology. It's well known exactly how it's caused. So that was a case of using mathematics to understand something that you see in the real world. And it helps you understand that when this happens, you don't necessarily have to be worried that your fridge broke. And you know that there are actually what you know, the policy implication is that over years, you've accumulated a pool of people much older than you were used to who can catch measles because you didn't vaccinate them. You've vaccinated something like 85% of the population, and each year you're accumulating 15 or 10% or whatever, and you have to find those older people and vaccinate those. That's taken too long, but never mind. Next example is much later. So I did this work, and actually what I ended up thinking was, oh, this is amazing. How does vaccination work? That's a great question. Really good dynamical systems questions, because you've got a highly nonlinear system inside each one of us. I was also in the process of this, there's really not much data in epidemiology. This is in the olden days. So I got very, very interested in quantifying and modeling the immune response. And round about the time I was publishing this, HIV was first described. And in the search for HIV vaccines, there was a lot of data. There was a lot of data about how much infectious virus, well, how many viruses you could find in people's blood, about the responding T cells, so the white blood cells that are part of the immune response. So we could get a lot of data about the sizes of the populations, but it was very hard to get any parameter estimates for actually what were those immune cells doing. So effectively, the thing we really wanted to know was to what extent does a good immune response shorten the life of an infected cell? And we were really struggling with those sorts of parameters until this kind of data started to come out. So now, this is still time, but whereas that was in years, this is in days. And this is now, let's call it the percentage of virus copies of different types. Well, actually, let's say the percentage of virus copies that are what are called escape mutants. So this is a mixture of ecology and evolution. Because what happens during individual HIV infections is that the immune response drives the evolution of types of virus that person's immune response cannot possibly see. So it's to do with details of how your immune response works. And this beautiful, beautiful data was collected, some from people who were quite newly infected, that showed you could match this, you could map this really quite specifically from early on in an infection, didn't really matter when, well, actually from absent, sorry, so this is 0 to 100% here, and that you could plot through time the outgrowth of the escape mutant. So, and it follows a beautifully classical sigmoid curve like that. But because we knew how long this took, basically from starting to being 100%, this was something like 180 days, let's say. So, and if I then tell you that basically the cycle time of an infected cell, basically the generation time going from one infected cell to the next infected cell inside a human is about a day, then what you can see is it takes 180 generations for the escape mutant, so the thing whose advantage is that it can't be seen by the immune system, to go from absent to present. And without showing you the equations, actually the equations are very simple, you can see, well, hang on a minute, that means its fitness advantage is actually quite small. It takes 180 cycles for it to become the dominant thing. That means the fitness advantage is small, and that means either the fitness cost is huge, or the immune response is not actually doing very much. And later on in other data, we were able to show that actually when these things transmit into another person, they don't actually disappear very fast. So the fitness cost is not huge. And the implication of that is that this bit of the immune response is actually, it's consistent, it's good enough to drive out a mutant, but it's not absolutely massive. And that was a huge surprise because there were very, very large numbers of these cells. But from this kind of evolutionary data, we were able to calculate that they were not actually having a huge impact on the lifespan of an infected cell. And what were the implications of that? I think really an understanding of why T-cell vaccines, so vaccines that elicited exactly this kind of response in beautiful numbers, gave the vaccine to the person, measured the immune response, all sorts of cells there, but not good, strong immunity. So those are two examples, a bit of epidemiology, a bit of within-host evolution. Let me finish by talking a bit about why policy and why engineering biology. So throughout all of this, I was working in an environment where lots of people were involved in science advice for government policy. The person who was my sort of second supervisor during this work actually had the job I have now. He was a man called Bob May. He was government chief scientific advisor. So I was in this environment where we saw the people we greatly admired doing this kind of work, and were able to be involved in various different sorts of advisory roles. So all our government departments have science advisors. Most of them have either lists of experts they can turn to or councils of experts who get to know them. So quite early in my career, I was able to take part in that kind of work and realised that I just found it tremendously interesting. And was asked, actually, in fact, I think by my predecessor, Patrick Vallance, early in his time in this role, to help write a report about which were the most important technologies for the United Kingdom to pursue. And the three that were chosen were, at that stage, AI, quantum, and engineering biology. So I wrote the engineering biology chapter for that, not because I'm an engineering biologist, but because I knew a bit about government by then, compared with most outsiders, quite a bit, to be honest. And also I knew about biology, I knew about quantification in biology. And I think the important thing was that I knew enough to be able to do a bit of translation and think carefully about, well, for my government friends, what is it they need to know? Because they don't actually need to know everything, and since they have to know a lot of things in a fairly shallow way, the trick is to decide, well, if we're going to tell them a few things about engineering biology, which ones should those be? So this was something called Science Capability Review that in the end must have come out, I don't know, about 2016, I would guess. I wrote the first version of the engineering biology, a bit of that, and eventually, when was it? Trying to think when. About 2020, the UK chose 5 critical technologies, of which those were 3, and then we added in, what do we call it? We call it next generation telecoms. Actually, what we really mean is telecommunications everywhere, and the other one is semiconductors. So that still stays a big focus for us in the UK. It's a technology where we very much hope we can continue to grow our own capability and both remain being at the forefront of of the new science, and also start to really spin out an industry that will help replace the industries that we will lose in the UK as we move away from extracting fossil fuels. So I thought I would stop there. Very happy to take any questions. I mean, to be honest, this is all quite a long time ago. If I can remember, I'll answer any questions about that. But I'd be particularly interested to hear from people who might be interested in science advice for policy, which is really what I do all day, every day now. Thank you very much. If you ask a question, I will repeat it, but that's just so that we can be sure it gets picked up by the camera. Yes?
So I was actually hoping that you'd be willing to share your cliff notes on what exactly the government officials would like to know out of all the information you can give them as a scientist.
That's a great question. So the question is, what would government officials like to know out of all the information they could be given? That's a great question. I think, first of all, predicting the future is a fool's game, but you can do it better than a government official can. I think real help with what's coming in the future. And I think, bear in mind, I think the thing that comes in the cliff notes about this is you need to decide, are you going to go in there and be a so-called honest broker, or are you going to go in there and be an advocate? And either is fine, but you must be clear in your own head which role you're playing. Because I say to my government colleagues, when someone's talking to you, to try to figure out what are they doing. Have they come because there's something they're passionate about and they want to push, or have they come because they're trying to help you see both sides of a complicated argument? I think the other thing, so that's one thing, help them think about the future. And then I think the other thing is actually, in a way, help them understand the corpus of scientific knowledge. I didn't talk about it, but another piece of work that I've done with a dear friend and colleague, Charles Godfrey, is what we call the Restatements Project, which is not about trying to find out new things, it's about just trying to write down in a sort of fair way, here is what we know. We did it first for tuberculosis in cows in Britain. Tuberculosis in cows in Britain is a big, long, enduring problem, and it's very, very controversial because it basically, the only way we have to control it at the moment is to kill badgers. Badgers are our largest, it's our largest bit of charismatic megafauna. It's kind of sad. I mean, well, we killed everything else. So it's a controversial issue and very heated debate, data that's sort of not as good as you'd like it. So I would say that's another thing that experts can do, is lay out, here's the evidence in some particular question, here's what's agreed, here's what's disagreed. It's very helpful to explain to government officials why things are disagreed. Because you can imagine, if you're coming from way outside a subject, that's a very hard thing to get your head around. Great question, thank you. Yes.
I have a science question.
Good.
If you don't mind.
Good.
So you said that it takes 180 days and 180 generations of the T cells, I
Yeah.
suppose, to make the next wave. With COVID, however, like alpha, beta, gamma, delta,
Yeah.
became much shorter. What was the reason
Shorter?
for that? I think
With
less than 180 days.
So this is inside an individual, remember?
Right.
So this is inside one person. So now you're talking about the waves
Population.
amongst the population.
Yes.
Yes. COVID was very new in humans, I would say, is probably what's going on there. So remember, this would be I'm just trying to think, when was this data? This was probably late 1990s. So by this stage, HIV had been in people for, 40 years about. So what you're talking about, remember, we saw COVID in its first few years in humans. So that's what I would guess was going on, is that, I would also guess that, we know that the immune response to an individual, the immune response was doing quite a lot. I would say that there are two differences there. I would say the main one is that the COVID immune response was doing plenty. But the second would be, I might expect that there would be by the time this was happening in humans, a whole lot of variants had already gone. So you were seeing, you could think of it as like sort of succession on the changes that you see very early on, I would expect them to happen faster. And then those things to just go away completely. Great question. Right, yes. I forgot to answer the question. Yes.
So, what did you feel the government needed to know about engineering?
Oh, yeah, great question. What did I feel the government needed to know about engineering biology? What you could do with it, I think, is the main thing. So I think one of the main things that it is important for government to understand about engineering biology is that it's not just a question of vaccines and pharmaceuticals. So I think that active imagination to see that by modifying genomes, we really can bend many, many materials to our will to be a better version of the natural product for a particular problem. So I would say that was the big message. We did also, I don't remember, I think at that stage we just sort of stated, we know we're good at this in the UK. At that stage we didn't have good data about that. These days we do, actually. So within Go Science we have a whole unit whose job it is to assess technologies in terms of a whole bunch of things like how many papers are there and how good are they, what's our patent productivity like, are we generating spin-outs, are we generating growth companies. So these days we have that data and we could say, look, we're good at the first 3 of those and terrible at the 4th. But we didn't have that information back then, so we just boldly stated, we know we're good at it.
Did you feel you were acting as an honest broker or advocate?
That's a great question. That's a great question. I think probably more advocacy, because we didn't have a broad, it was not based on, oh look, here's a survey of all the technologies we could pick as a critical technology, and therefore you would pick this one. So no, I think that's a reasonable question. Yes?
How did you happen to advise policymakers?
Happen to, was that word?
Yeah, how did it come to?
Yes. So I was probably asked the first time. I think my first science advisory role was advising the ministry, no, it's already called the Farming Ministry, so the Department for the Environment, Farming and Rural Affairs, DEFRA. And I was invited to join their Science Advisory Council by the chair of that council, and I expect he was told by somebody else, you should ask Angela to apply. So I had to apply. It was a formal process. You know, we have a thing called Nolan Rules in the UK. But I don't think it wouldn't have crossed my mind to apply if I hadn't been asked. I do spend a lot of time now telling people in the UK about these roles. They're not full-time roles, so I would go sort of 4 times a year for 2 days into DEFRA to talk to them. The main thing I did whilst I was on that group was to talk to DEFRA about how they could use risk analysis to bridge between their own scientists, because they had a lot of very deep pockets of expertise, but to make a language that could be shared between the scientists who were doing the science and the policymakers who are having to make decisions that should be using that science. And I think that's really the reason risk analysis is actually rather powerful, is it lets people who are making decisions use science to think about these big questions like, well, what are the worst things that might happen, and what do I even mean by worst? So I think the slightly depressing answer to that, but I will be honest with you, is I was invited to apply. Yes?
Now, I like it a lot that in your title when you separated mathematical biology and engineering biology. So you mentioned language barrier, but as you know, there's a language barrier even within the science, leave aside the government. Mathematical biology, some places they don't even separate those. I would like you to tell us from the side of government official now, how do you see like this Oxford example, how this mathematical biologists and engineering biologists interaction can be optimized? Because this is an important question also for OIST. If you remember OIST, we really have mathematical biologists and engineering biologists, but there's not much interaction. Of course, we work on that. So what would be your advice from taking like experience of Oxford?
Okay. So the question is, how shall we, let's be careful with the language about mathematical biology and engineering biology, and what should the interaction be. And I think I would see them as overlapping. You can do bits of mathematical biology that have no engineering biology in them, and you can do bits of engineering biology. I don't know, I think I would find it odd if you were doing engineering biology that really had no mathematics in it at all. I guess you could, probably. Because we in the UK use engineering biology quite loosely. So really what we mean is, our definition is, I haven't got it written down, but our definition is something like using engineering principles to make designed biological products. So for example, we don't insist in our definition that there should be genetic modification. And in fact, many of our examples have no genetic modification. So yes, you could have, so it's an overlap, not one inside the other. That would be the main thing I would say, is there's an overlap between them, but they're two separate things. So there was no engineering biology in that. There's no engineering biology in all the mathematical epidemiology that we all did for COVID. There was nothing got engineered there. So I think they're both broad, and I wouldn't say either was subsumed into the other.
As governments come and go quite rapidly, or politicians change roles, do you see a sudden shift in priorities, and how do you cope with, say, a sudden shift when making policy or advising policy decisions?
Patiently. Yeah, no, that's a great question. I mean, one of the things that's happened in the UK that actually is a really terrific thing is that Patrick Vallance, who held my job until I took it, has actually come back into government as minister. And that has given us real continuity of language. So, for example, although we are not long-term committed to what our priority technologies will be, the new government recognises the 5 critical technologies and hasn't dropped them. Now, since the whole point of having a chosen set of critical technologies is to have some focus amongst all the things we could do, which one should we focus on? We are immensely relieved to have that continuity of purpose. I think it's one of the reasons that being at the beginning of a government that we're pretty sure will last for 5 years, that has been very clear what its missions will be, what is it going to try to change, is an enormous relief to us. In that we know we can put structures in place, we can make decisions, we can start to gather advice, some of which will take a year or so on its own, knowing that that piece of purpose will still be there. When government priorities change, sometimes you just have to, you don't tear it up, but sometimes you have to take the piece of work you were doing and put it away in a drawer. We were talking about this, who were we talking about this with? Anyway, you don't tear it up because the right idea will usually come back into fashion again. But, you know, I mean, I'm a civil servant and the word is meaningful. I don't get to say, you can't do that, no matter how bad an idea I think it is. Well, I can try.
So, I see, I have a final question for you.
Yes.
And that is, I saw a survey recently among, I think, 17 countries in the world, among what public really, believe in that scientists will solve the problems in the world. And it was quite astonishing, maybe some of you have seen this survey, that really the countries that believe in scientists are not the countries that usually have worked a lot with science, like Europe and the United States. We're quite low, and public don't believe, and it's going down. Mm-hmm. On the other hand, in India and China and Japan, actually it's quite strong. So what is how can that be when the countries that really have a mature science education and also research, how come we see this mistrust coming in public? And what does that mean for the government's way of handling the situation?
So I think we should look at ourselves. I think if we are not trusted, we should ask, what have we done that was untrustworthy? And I think in the UK, although it's a long time ago, we still are very anxious about what happened with BSE. So BSE was the disease of cows, called mad cow disease. And we scientists, I wasn't there, but I will share the responsibility, allowed government ministers, and we should not have allowed this, to stand up and say British beef is safe at a time when British beef was not safe. And that has cost us terribly in trust, and I don't think that's unreasonable. And then we come back to your question, what should we have done? And at some stage, somebody should have said, Minister, you cannot go on the record and say that, because think what will happen to your reputation should there ever be any cases of this disease in people. And of course, by 1996, we knew there were cases of this disease in people. So I think maybe somebody should have sacrificed their career, because that might have been a career-destroying move. But given that we knew we didn't know, there were arguments by analogy that it would be safe. Humans have eaten the equivalent pathogen in sheep for thousands of years, and we see no trace of any disease from humans from eating the sheep thing. But in biology, analogy often fails, and we should have been more thoughtful about that.
Admit also.
Yeah, yeah.
Yeah, so of course, this also comes to the maybe the thinking of what do we need to do, because these big problems we have are global. So
Yes.
somehow we need to actually make public to also believe in science, and science is changing all the time.
Yes.
So I guess
Yes.
is there a solution there?
So I think that does require very clear discussion amongst civil servants, because remember, certainly in the United Kingdom, most civil servants have no science background. Not little, but none. They've last done science when they were 16. So discussion about uncertainty amongst scientists, and a difficult discussion between science, the scientific civil service, and politics about uncertainty, which scientists love. It's our bread and butter, after all. If there was no uncertainty, we'd just all have to be engineers, which is marvellous, but you know, your job is to find uncertainty and try to resolve it, yes. Politicians hate uncertainty because it's what makes policies fail. So that conversation, I think, is actually, of course, very rich. Should be a very rich, powerful conversation, because what we should be able to do is warn politicians of the unresolved uncertainties and help them think about what are the implications of that. And that will often take you away from natural science, first into behavioural sciences, and then also into other tools, other much more narrative tools that we would call futures tools. So where you say, we don't know what will happen, but we could be quite thoughtful and systematic about the various things that could happen and allow you to think through what their implications would be. So we should have done a futures exercise where we said, what if it turns out that BSE does infect people? How bad are you going to look, Mr. Selwyn Gummer, having made your daughter eat that hamburger? Because that is an iconic piece of newsreel for the United Kingdom. Turns out she was crying. The poor child was crying because the hamburger was too hot.
Oh, well, a lot of things to do, really.
Lots to do.
Yes. Thank you very much, and please give a hand for
Thank you, Karen. Now I lost my water. Oh well, I'll take one of these. I know I can see my water. Great question. Thank you.